In this segment, we will learn about a connection which was once considered extremely rare. The connection between cancer and genetics, or in other words, a genetic predisposition to develop cancer throughout life. The cause for cancer was not known for centuries. In the 20th century, it was linked mostly to external stimuli, such as radiation and smoking, leading to the term carcinogens. In a landmark paper in 1971, Alfred Knudsen proposed the two-hit hypothesis, in which the initial hit, or genetic event, occurs in the germline. This was described in retinoblastoma, a rare tumor of the eye occurring in very young children. Here, you can see the white reflection in the eye of a newborn infant, suggesting infiltration of the retina by malignant cells. About 40% of patients with retinoblastoma have family members with a history of this disease. A deletion or a mutation in chromosome 13 in the locus of the RB gene was identified in these patients. The initial hit was inherited and the second occurred in young infancy. Indeed, these patients not only present at a young age, but also often have multiple tumors in the eyes. In this slide, you can see an example of a pedigree of a family with retinoblastoma due to a mutation in the RB1 gene. Since this is not a complete deletion of the gene, and since an additional mutation event is needed, there is incomplete penetrance. Thus, some family members carry the mutations but did not develop retinoblastoma, as depicted in the dashed circles and squares. The mutation was found in exon 19 of the RB1 gene, with an alteration of guanine to thymidine, as shown in the lower panel. Around the same time, Frederick Lee and Joseph Fraumeni noticed that some families have increased cancer susceptibility, not only one kind of cancer, but of many. This is an example of a pedigree in which family members had sarcoma, breast cancer, brain tumors, other tumors, or a combination. The Lee-Fraumeni syndrome was defined as a proband with sarcoma younger than 45 years of age, who has a first-degree relative with any cancer below 45 years, plus another first or second-degree relative with either any cancer below 45 years or a sarcoma at any age. This definition has loosened with time, given the identification of many other cancers, such as adrenocortical carcinoma or brain tumors in this syndrome, and mostly with the understanding of its genetic background. In 1990, David Malkin identified germline TP53 mutations in patients with Lee-Fraumeni syndrome. This is a very important gene, referred to as the guardian of the genome, since it is capable of sensing stress signals coming from DNA damage or UV radiation, as depicted in this image, and protects these damaged cells by controlling their growth, DNA repair, and apoptosis. Here, you can observe the frequency of germline mutations in the P53 gene, with the majority of mutations in the DNA binding area in blue, or in the tetramerization domain in green. Due to the importance of this protein, there are several databases which are dedicated to its biology, which you will get to know in the practice session. These findings revolutionize the knowledge regarding the genetic background of cancer, especially childhood cancer. Currently, detailed family history is taken from every patient with cancer. Patients with either sarcoma at a young age, who have family members with other malignancies, patients with a rare form of a tumor in the adrenal gland, patients with more than one malignancies, and others should be referred to genetic testing and counseling. Several family cancer syndromes caused by other genes, such as the BRCA gene, have been identified. But the significance of knowing the patients with predisposition is not only limited to pure scientific curiosity. It also has potential implications. First is the option of pre-gestational diagnosis. Through in vitro fertilization, or IVF, geneticists can test one of the 8 or 16 cells of the very early embryo and discover whether it carries the normal gene or the mutated copy. This is depicted in this image of 5 potential embryos. Analyzing one cell of each, the physicians can identify that 3 are affected, colored in red, and 2 are unaffected, colored in green. Then, only embryos not carrying this potentially fatal gene can be used for further implantation and will hopefully lead to a normal pregnancy. A second intervention is through screening programs. The group from the Hospital of Sick Children in Toronto, led by Dr. Malkin, pioneered this in patients with Li-Fraumeni syndrome. By routine total body MRI, the tumors that developed in these patients were identified in smaller sizes and less malignant forms before metastasis occurred, and they were removed surgically, leading to improved survival of patients screened. In this graph, you can see that all patients in the screening program, represented by the red line, survived the follow-up period, more than 60 months, without events of death, while patients who declined screening for various reasons had increased mortality, as seen in the blue line. This work led to the foundation of cancer predisposition clinics in many pediatric oncology departments, with goals of identifying patients with germline mutations causing a risk for cancer development, and ongoing screening and detection in patients at risk. A similar project is now operating at our hemato-oncology department at the Safra Children's Hospital in the Sheba Medical Center. In this project, children with predisposition to various cancers due to germline mutations in known cancer genes, such as p53 and mismatch repair genes, are being surveyed intensively. In some cases, indeed, early detection of tumors allowed surgical removal and definitely improved the long-term outcomes. During the practice session, we will get to know useful resources relevant to familial cancers. Maya and Ronen, as you see, the identification of people with the predisposition to cancer already yields prognostic benefits. As our understanding of the interplay between genetic predisposition and subsequent somatic mutations increases, we will be able to detect cancer early and treat it better. In the next segment, we will discuss the emerging field of immunotherapy that has risen due to better understanding of how our immune system interacts with tumors.